The clinical mislead. The first tremor is not the beginning. The patient sits in a neurology clinic with a subtle tremor in the right hand that appears only at rest. When the hand is placed in the lap, the fingers move rhythmically in a slow, pill-rolling motion. When the patient reaches for a cup, the tremor briefly diminishes. Strength is intact. Reflexes are normal. Gait shows only slight reduction in arm swing. The face appears mildly less expressive than before, but speech is clear. The diagnosis offered is early Parkinson's disease. The implication is that the disease has just begun. It has not. By the time a resting tremor becomes visible, degeneration inside the midbrain has already progressed for years. Parkinson's disease begins in dopaminergic neurons of the substantia nigra pars compacta, not in the hand. The tremor is the threshold event, the moment when compensatory mechanisms inside the basal ganglia network can no longer stabilize motor output. The substantia nigra is a small pigmented structure in the midbrain. Its neurons project densely into the striatum, releasing dopamine that modulates motor circuit balance. These neurons do not fail suddenly. They undergo prolonged metabolic stress, synaptic dysfunction, and progressive loss long before motor signs emerge. Clinical detection occurs only after a substantial percentage of dopaminergic neurons have already died. Motor symptoms require crossing a biological threshold. Studies of postmortem brains show that by the time classic motor features appear, approximately 50 to 70% of dopaminergic neurons in the substantia nigra are already lost. Striatal dopamine concentration has dropped to a fraction of its baseline level. The visible tremor is therefore not the beginning of disease. It is the consequence of advanced network imbalance. Early degeneration does not produce immediate collapse because the basal ganglia possess buffering capacity. Surviving dopaminergic neurons increase dopamine turnover. Postsynaptic receptors become more sensitive. Firing patterns adapt to preserve movement fluidity. The network compensates silently. Motor performance remains functionally intact. This compensation creates the clinical illusion of late onset. Patients often recall subtle changes years before diagnosis. Reduced sense of smell, constipation, vivid dreams with physical enactment during sleep, mild fatigue, or decreased facial expressivity. These are not random symptoms. They reflect earlier involvement of non-motor dopamine-related and brainstem circuits preceding overt motor collapse. When tremor first appears, the patient and clinician perceive a beginning. In reality, the disease has already progressed through molecular, synaptic, and neuronal phases of degeneration. The motor system simply reaches a tipping point where inhibition and activation within basal ganglia loops become unbalanced. Resting tremor itself is not produced directly by dopamine deficiency alone. It emerges from oscillatory instability within thalamocortical circuits when inhibitory control from basal ganglia becomes dysregulated. This oscillation represents network desynchronization, not a simple chemical shortage. Bradykinesia, the slowness of movement, often precedes tremor. Patients may notice reduced arm swing or difficulty initiating motion. These changes are subtle and easily attributed to aging. They represent early imbalance between direct and indirect motor pathways, long before overt shaking commands attention. Rigidity develops as increased inhibitory output from the globus pallidus interna suppresses thalamic activation of motor cortex. Muscles are not weak. They are under continuous inhibitory modulation that restricts fluid movement. This too reflects circuit imbalance, not muscular pathology. The first tremor is therefore a surface signal of deep structural loss. It is the moment at which compensation fails to mask degeneration. The dopamine network has been collapsing gradually. The visible sign simply marks the point at which remaining neurons can no longer maintain equilibrium. Parkinson's disease does not begin in the clinic. It begins years earlier in the midbrain, at the level of mitochondrial stress and protein misfolding inside vulnerable dopaminergic neurons. The tremor is not the origin. It is the late-stage announcement that a hidden collapse has already progressed beyond silent containment. Basal ganglia architecture, the dopamine network explained. To understand early dopamine network collapse, the architecture of the basal ganglia must be precise. Movement is not generated solely in motor cortex. It is selected, filtered, and modulated by a deep subcortical network composed of the striatum, globus pallidus, subthalamic nucleus, and substantia nigra. Dopamine does not initiate movement directly. It modulates how this network balances activation and inhibition. The substantia nigra pars compacta contains dopaminergic neurons, whose axons project densely to the striatum, which includes the caudate nucleus and putamen. The putamen is particularly central to motor control. Within the striatum, dopamine acts on two major populations of medium spiny neurons distinguished by receptor type. D1 receptor-expressing neurons forming the direct pathway, and D2 receptor-expressing neurons forming the indirect pathway. The direct pathway facilitates movement. When activated, it reduces inhibitory output from the globus pallidus interna to the thalamus, allowing the thalamus to stimulate motor cortex more effectively. The indirect pathway suppresses movement. It increases inhibitory output through a multisynaptic route involving the globus pallidus externa and subthalamic nucleus, ultimately restraining thalamic activation of cortex. Dopamine's role is modulatory and asymmetric. Dopamine excites D1 neurons in the direct pathway, promoting movement. Simultaneously, it inhibits D2 neurons in the indirect pathway, reducing movement suppression. The combined effect is facilitation of voluntary motor activity with flexibility and fluidity. In a healthy brain, this dual modulation maintains dynamic balance. Motor commands are not simply turned on or off. They are fine-tuned. The basal ganglia allow appropriate movements to proceed while suppressing competing or unnecessary actions. This filtering function depends critically on intact dopaminergic signaling. When dopamine levels decline gradually, the balance shifts. Direct pathway activation weakens because D1 stimulation falls. Indirect pathway inhibition weakens as well because D2 suppression decreases. The net effect is increased inhibitory output from the globus pallidus interna to the thalamus. Thalamocortical stimulation declines. Movement initiation becomes difficult. Importantly, this imbalance does not immediately produce tremor. Early in degeneration, residual dopamine still modulates enough receptors to preserve functional movement. Network plasticity allows partial compensation. Surviving dopaminergic neurons increase firing rate. Dopamine synthesis and release per neuron increase temporarily. The basal ganglia network is not static circuitry. It operates through oscillatory patterns. Cortical signals enter the striatum, are processed through direct and indirect pathways, and return to cortex via the thalamus in continuous loops. These loops control not only movement amplitude, but timing and initiation. Dopamine stabilizes these loops, preventing excessive inhibitory oscillation. As dopaminergic neurons die, the indirect pathway becomes progressively overactive relative to the direct pathway. The subthalamic nucleus becomes hyperactive. The globus pallidus interna increases tonic inhibition of the thalamus. Cortical motor areas receive less excitatory feedback. The result is bradykinesia, the slowness that characterizes Parkinson's disease. The tremor component arises from abnormal rhythmic firing within thalamocortical circuits that become unstable when basal ganglia output loses modulatory precision. This instability reflects network-level dysregulation, not isolated muscle pathology. The basal ganglia architecture also extends beyond motor control. Associative loops connect prefrontal cortex to striatum, influencing executive function. Limbic loops connect emotional circuits. Dopamine depletion therefore affects more than movement, though motor symptoms dominate early recognition. The key structural vulnerability lies in the dopaminergic projection from substantia nigra to striatum. These neurons are uniquely susceptible to oxidative stress due to high metabolic demand and dopamine's intrinsic biochemical properties, which can generate reactive oxygen species during metabolism. When degeneration begins, the basal ganglia network does not collapse instantly. It shifts gradually toward inhibitory dominance. Direct pathway underactivation and indirect pathway overactivation create a system bias toward suppression rather than facilitation of movement. Understanding this architecture clarifies why early Parkinson's disease is a network disorder before it is a motor disorder. Dopamine network collapse refers to the destabilization of this delicate balance. The visible tremor and rigidity represent the final expression of deeper circuit imbalance that has been evolving silently within basal ganglia loops. The dopamine network is therefore not a simple chemical system. It is a distributed modulatory framework that stabilizes motor selection. When its integrity declines, the collapse manifests first as subtle slowing and loss of automatic movement, long before overt shaking commands attention. Early dopaminergic stress, before neurons die. Parkinson's disease does not begin with sudden neuronal death. It begins with stress inside dopaminergic neurons that makes them progressively unstable long before they disappear. The earliest phase of degeneration is molecular and synaptic, not structural. Dopaminergic neurons of the substantia nigra pars compacta are uniquely vulnerable because of their physiology. They are autonomous pacemakers. Even at rest, they fire continuously to maintain baseline dopamine tone in the striatum. This constant activity requires sustained calcium influx through voltage-gated channels. Chronic calcium entry increases mitochondrial workload and metabolic demand. Mitochondria are central to early collapse. In Parkinson's disease, mitochondrial function becomes impaired through genetic susceptibility, environmental factors, or cumulative oxidative stress. Complex I dysfunction reduces ATP production efficiency. As energy generation declines, reactive oxygen species increase. Dopamine metabolism itself produces oxidative byproducts, further amplifying cellular stress. Before neurons die, synapses begin to fail. Dopamine release into the striatum becomes inconsistent. Phasic bursts that normally encode movement initiation weaken. Baseline tonic dopamine levels fluctuate. Striatal median spiny neurons receive less stable modulation. Alpha-Synuclein misfolding is a central molecular event in this early phase. Normally a presynaptic protein involved in vesicle regulation, Alpha-Synuclein begins to aggregate abnormally. Misfolded protein accumulates within neuronal cytoplasm and synaptic terminals. These aggregates interfere with vesicle trafficking and neurotransmitter release. Synaptic terminals in the striatum may degenerate before the cell bodies in the substantia nigra are visibly lost. This means dopamine depletion at the synapse can precede measurable neuronal death in the midbrain. Function declines before anatomy collapses. This synaptic dysfunction explains subtle early motor changes. Reduced dopamine signaling weakens direct pathway facilitation. The patient may notice decreased automatic movements, less arm swing, slower handwriting, reduced facial expression. These changes are mild because remaining neurons compensate. Surviving dopaminergic neurons increase dopamine synthesis and turnover to offset loss. Dopamine transporters adjust reuptake dynamics. Postsynaptic receptors increase sensitivity. These compensatory mechanisms maintain motor output temporarily, masking degeneration. However, compensation has metabolic cost. Increased firing rate and dopamine turnover accelerate oxidative stress. The remaining neurons work harder, increasing vulnerability. This accelerates the degenerative cycle. Inflammatory processes also begin early. Microglia in the substantia nigra become activated in response to misfolded protein and cellular stress. Chronic neuroinflammation contributes to oxidative damage and neuronal fragility. The environment becomes progressively hostile to dopaminergic survival. Importantly, Early degeneration is regionally selective. The ventrolateral tier of the substantia nigra, which projects to the putamen, is particularly vulnerable. This explains why motor symptoms eventually dominate. Associative and limbic projections may be affected later. During this early phase, no tremor is visible. No rigidity is apparent. The dopamine network is destabilizing internally, but output remains within functional limits. The system appears intact externally because compensatory adjustments buffer decline. The fear embedded in early Parkinson's disease lies in this invisible stage. By the time neurons are lost in sufficient numbers to produce tremor, synaptic dysfunction and metabolic stress have been ongoing for years. The disease begins long before diagnosis. Early dopamine network collapse is therefore a progression. Mitochondrial inefficiency, oxidative stress, alpha-synuclein aggregation, synaptic dysfunction, increased compensatory firing, and eventual neuronal death. The tremor does not signal the start of this cascade. It signals that compensation has failed. Before neurons die, they struggle. Before tremor appears, dopamine signaling has already destabilized. The collapse begins silently at the level of energy metabolism and protein folding, long before the hand begins to shake. The compensation phase, why symptoms are delayed. Parkinson's disease progresses for years before it becomes clinically obvious because the dopamine network is built with reserve capacity. The brain does not rely on a single neuron for movement modulation. Thousands of dopaminergic neurons project from the substantia nigra to the striatum. Early loss can be masked by adaptive mechanisms within surviving cells and downstream circuits. When dopaminergic neurons begin to die, the remaining neurons do not remain passive. They increase their firing rate. Dopamine synthesis per neuron rises. Vesicular packaging becomes more efficient. Dopamine turnover accelerates. This compensatory upregulation temporarily stabilizes extracellular dopamine levels in the striatum despite ongoing cell loss. At the postsynaptic level, receptor sensitivity changes. D1 and D2 receptors on medium spiny neurons become more responsive to available dopamine. Even reduced dopamine release can produce sufficient downstream activation because receptor density and signaling efficiency increase. This is receptor supersensitivity. Dopamine transporter dynamics also adjust. Reuptake efficiency may decrease, allowing dopamine to remain longer in the synaptic cleft. This prolongs signaling despite lower overall release. These biochemical adjustments maintain basal ganglia balance during early degeneration. Network plasticity contributes further compensation. Cortical motor areas may increase excitatory drive to the striatum. Thalamocortical loops adapt firing patterns to preserve movement fluidity. The system reorganizes to maintain function within narrower margins. This compensation explains why motor symptoms emerge late relative to neural loss. Studies consistently demonstrate that clinical diagnosis typically occurs after more than half of nigral dopaminergic neurons are already gone. The brain masked that loss until thresholds were crossed. However, compensation is not neutral. Increased firing rate increases calcium influx. Elevated dopamine turnover increases oxidative stress. Mitochondrial burden intensifies. The very mechanisms that preserve function accelerate degeneration of surviving neurons. This creates a paradox. The network appears stable externally, but internally it is under escalating strain. Surviving neurons are operating beyond physiological baseline to maintain dopamine tone. Over time, this strain becomes unsustainable. Subtle early signs may appear during this phase. Movements that require rapid modulation become less fluid. Automatic behaviors, such as arm swing or facial expression, decrease because automatic motor loops are more sensitive to dopamine fluctuation than consciously driven movements. The patient may not notice these changes immediately. Non-motor symptoms often precede motor threshold crossing during this phase. Loss of smell reflects early involvement of olfactory pathways. Constipation reflects autonomic network involvement. REM sleep behavior disorder reflects dysfunction in brainstem circuits. These signs emerge because compensation in non-motor circuits may be less robust. Eventually, dopaminergic neuron loss reaches a level where compensation fails abruptly. When striatal dopamine concentration falls below approximately 20 to 30% of normal, the indirect pathway becomes dominant. Inhibitory output from the globus pallidus interna increases persistently. Motor cortex stimulation declines measurably. This is the threshold crossing. Tremor, bradykinesia, and rigidity become visible, not because degeneration suddenly accelerated, but because compensatory capacity has been exhausted. The compensation phase is therefore deceptive. It delays diagnosis. It creates the illusion of late onset. It hides structural loss beneath functional adaptation. By the time the tremor appears, the network has been struggling silently for years. Early dopamine network collapse is not sudden failure. It is prolonged compensation followed by structural tipping point. The visible symptom is the end of buffering, not the beginning of disease. Threshold crossing, when tremor and bradykinesia appear. There is a biological threshold in Parkinson's disease at which compensation collapses and motor symptoms emerge. This threshold is not subjective. It corresponds to measurable structural loss within the substantia nigra and measurable depletion of dopamine in the striatum. By the time bradykinesia becomes clinically evident, a substantial proportion of dopaminergic neurons in the substantia nigra pars compacta have degenerated. Striatal dopamine concentration has fallen to a fraction of its original baseline, reduced to a small fraction of baseline levels. The buffering mechanisms that once masked decline can no longer sustain motor balance. The direct pathway, which facilitates movement through D1 receptor activation, is now understimulated. The indirect pathway, normally restrained by dopamine acting on D2 receptors, becomes relatively overactive. The subthalamic nucleus increases excitatory drive to the globus pallidus interna. Inhibitory output to the thalamus rises persistently. Thalamocortical stimulation weakens. Bradykinesia emerges from this imbalance. It is not simple slowness due to muscle weakness. It is impaired initiation and reduced amplitude of movement because cortical motor areas receive diminished facilitation. The patient intends to move, but the motor program is not released efficiently. Rigidity reflects continuous overactivity of inhibitory basal ganglia output. Muscles are held in increased tone because inhibitory modulation of movement circuits is excessive. Unlike spasticity caused by corticospinal injury, rigidity in Parkinson's disease is uniform throughout the range of motion, reflecting subcortical modulation imbalance. Resting tremor arises from oscillatory instability within thalamocortical circuits when dopamine-mediated stabilization is lost. The tremor is rhythmic, typically 4 to 6 Hz, and diminishes during voluntary movement because cortical activation temporarily overrides basal ganglia oscillation. Tremor is therefore not a direct effect of dopamine absence, but a network oscillation phenomenon triggered by imbalance. Importantly, threshold crossing is asymmetric initially. Parkinson's disease often begins on one side of the body because degeneration within the substantia nigra may be uneven. Unilateral dopamine depletion produces unilateral motor signs before bilateral involvement becomes apparent. The emergence of symptoms does not mean degeneration accelerates suddenly. It means structural loss has reached a critical density. The remaining dopaminergic neurons can no longer increase firing sufficiently to compensate. Receptor hypersensitivity cannot overcome inadequate dopamine supply. Network plasticity is exhausted. Once this threshold is crossed, progression becomes clinically visible. As more neurons degenerate, symptom severity increases. Bradykinesia worsens. Gait becomes shuffling. Postural reflexes decline. The motor system loses flexibility. At this stage, dopamine replacement therapy such as levodopa often produces dramatic improvement. This response confirms dopamine deficiency as a primary driver of motor symptoms. However, symptomatic improvement does not restore lost neurons. It temporarily compensates for biochemical deficit. Threshold crossing marks the transition from silent network strain to overt motor dysfunction. It reveals the extent of structural loss that has already occurred. The tremor and slowness are not early warnings. They are late indicators of substantial neuronal death. Parkinson's disease becomes visible when dopamine network collapse surpasses compensation. The hand tremor that prompts evaluation is not the beginning of degeneration. It is the signal that the system's reserve has failed and that structural loss is now functionally unavoidable. Network desynchronization, beta oscillation entrapment. When dopamine depletion crosses threshold, the basal ganglia do not simply become underactive. They become abnormally synchronized. The collapse is not only chemical, it is electrophysiological. In healthy motor circuits, neuronal firing patterns fluctuate dynamically. Signals flow through the cortex, striatum, globus pallidus, subthalamic nucleus, and thalamus in coordinated but flexible rhythms. Dopamine stabilizes this system, allowing rapid transitions between motor states. Movements are initiated smoothly, adjusted continuously, and terminated appropriately. As dopamine declines, the balance between direct and indirect pathways shifts toward excessive inhibition. The subthalamic nucleus becomes hyperactive. The globus pallidus interna increases tonic inhibitory output. Thalamocortical drive decreases. But beyond simple inhibition, oscillatory behavior changes. Electrophysiological recordings in Parkinson's disease reveal exaggerated beta band oscillations, typically in the 13 to 30 Hz range, within basal ganglia thalamocortical circuits. Beta oscillations are associated with maintenance of the current motor state. When they become excessively synchronized, the system becomes resistant to change. This state is often described as beta entrainment. Neural populations fire in overly synchronized rhythms, reducing flexibility. Movement initiation requires desynchronization. When beta activity remains elevated, initiation is delayed. Bradykinesia emerges not only from reduced dopamine, but from persistent oscillatory rigidity within the network. Deep brain stimulation of the subthalamic nucleus reduces beta oscillation amplitude. Clinical improvement correlates with reduction in abnormal synchronization. This demonstrates that motor symptoms are linked to pathological network rhythms, not solely neurotransmitter deficiency. Network desynchronization also explains freezing of gait. In advanced Parkinson's disease, sudden inability to initiate stepping occurs when motor circuits fail to transition from a stable to a dynamic state. Beta entrainment prevents flexible shifting. The feet appear stuck despite intention. The dopamine network normally modulates oscillatory balance. Loss of dopaminergic input destabilizes this regulation, allowing pathological rhythms to dominate. Surviving neurons fire in increasingly synchronized bursts rather than flexible patterns. Importantly, these oscillatory changes precede overt structural collapse in some circuits. Even before complete neuronal loss, altered dopamine signaling can disrupt firing patterns. This reinforces the concept that Parkinson's disease is a network disorder as much as a degenerative one. Thalamocortical loops become entrained in rigid rhythms. Motor cortex output loses variability. Movement amplitude decreases. Reaction time increases. The body appears slow and stiff, not because muscles are weak, but because neural timing is locked into maladaptive synchronization. As degeneration progresses, oscillatory abnormalities spread beyond motor circuits. Cognitive and limbic loops may also exhibit reduced flexibility, contributing to executive dysfunction and mood changes later in disease. The collapse of dopamine signaling, therefore, produces two parallel consequences: chemical deficiency and rhythmic entrapment. The first reduces modulation. The second reduces adaptability. When beta oscillations dominate, the motor system becomes resistant to change. Initiation requires overcoming persistent inhibitory synchronization. The patient must exert greater effort to begin movement, and once movement begins, it lacks fluidity. Early dopamine network collapse thus transitions from molecular stress to structural loss to oscillatory rigidity. By the time tremor and bradykinesia are visible, the network is not only dopamine deficient, it is rhythmically entrapped in patterns that suppress flexibility. This is not simply a shortage of dopamine. It is a collapse of timing architecture within basal ganglia circuits. And once entrainment is established, restoration of normal synchronization becomes increasingly difficult. Non-motor symptoms come first. The collapse begins outside movement. Parkinson's disease is classified as a movement disorder because tremor and bradykinesia are visible. However, degeneration often begins in regions unrelated to voluntary motor control. The dopamine network collapse is broader than the motor loop, and in many patients, non-motor symptoms precede tremor by years. Loss of smell is one of the earliest detectable signs. The olfactory bulb and anterior olfactory nucleus show alpha-synuclein pathology in early stages of the disease. Patients may notice reduced ability to detect odors long before a neurologist detects motor slowing. This is not incidental. It reflects early involvement of non-motor neural circuits vulnerable to protein aggregation. Constipation frequently appears years before diagnosis. The enteric nervous system contains dopaminergic and autonomic neurons susceptible to alpha-synuclein accumulation. Degeneration within autonomic pathways slows gastrointestinal motility. Because constipation is common in the general population, it is rarely linked to impending neurodegeneration. REM sleep behavior disorder is another prodromal feature. Normally, brainstem circuits suppress muscle tone during REM sleep, preventing physical enactment of dreams. In early Parkinson's disease, degeneration within pontine structures disrupts this suppression. Patients may act out vivid dreams years before motor symptoms emerge. This reflects early brainstem involvement. Mood changes, including depression and anxiety, may also precede motor signs. Dopaminergic projections to limbic and prefrontal regions modulate reward and emotional processing. Reduced dopamine tone in these circuits can produce motivational decline and affective disturbance independent of motor impairment. These early non-motor symptoms illustrate a critical principle. Parkinson's disease is not confined to the substantia nigra at onset. Braak staging proposes that alpha-synuclein pathology may begin in lower brainstem and peripheral autonomic structures before ascending to midbrain dopaminergic neurons. Whether this sequence is universal remains debated, but clinical observation supports early non-motor involvement. The prodromal phase can last a decade or more. During this time, dopaminergic neurons in the substantia nigra are already experiencing metabolic stress. Synaptic dysfunction is present. Compensation maintains motor stability. The patient may seek care for sleep disturbances or constipation without recognition of underlying neurodegeneration. By the time tremor appears, pathology has likely spread beyond isolated midbrain regions. The dopamine network collapse is not a sudden focal event. It is a distributed process affecting multiple neural systems sequentially. Recognition of prodromal symptoms is important not because it prevents structural loss retroactively, but because it clarifies that visible motor collapse is a late stage. The disease has been unfolding silently across networks long before shaken commands attention. Importantly, not every individual with constipation or REM behavior disorder will develop Parkinson's disease. These symptoms are nonspecific in isolation. What makes them significant is their presence in combination with subtle motor changes and risk factors. The non-motor phase reveals the hidden breadth of dopamine network collapse. It underscores that Parkinson's disease is a systems disorder involving motor, autonomic, sleep, and emotional circuits. The fear embedded in this phase is temporal. The earliest detectable symptoms are easily dismissed. By the time motor signs prompt evaluation, Neuronal loss has already surpassed compensation thresholds. Non-motor symptoms therefore function as early markers of underlying degeneration. They are not minor side features. They are evidence that dopamine and related neural networks are destabilizing before tremor ever appears. Parkinson's disease begins in silence outside movement. The hand tremor that defines it publicly is only the visible fragment of a broader network collapse that started years earlier in circuits most people would never suspect. Progression beyond motor executive and autonomic spread. As Parkinson's disease advances, degeneration extends beyond the motor loop of the basal ganglia. The dopamine network is not confined to the putamen and primary motor circuits. Dopaminergic projections also reach associative and limbic regions. When these circuits lose modulation, cognitive and autonomic systems begin to destabilize. The prefrontal cortex depends on dopaminergic input for executive function, working memory, and behavioral flexibility. Reduced dopamine tone in frontostriatal associative loops impairs planning, multitasking, and cognitive speed. Patients may describe difficulty organizing tasks or maintaining attention. This decline differs from Alzheimer-type memory loss. Encoding remains relatively preserved early, but retrieval and executive coordination weaken. The caudate nucleus, part of the dorsal striatum, plays a central role in cognitive loops. Dopamine depletion here disrupts communication between prefrontal cortex and thalamus. Executive inefficiency emerges gradually. Slowed processing becomes persistent. Complex decision-making requires more effort. Limbic circuits are also affected. Dopaminergic projections to the ventral striatum modulate reward and motivation. Reduced signaling in these pathways contributes to apathy, reduced initiative, and emotional blunting. Depression in Parkinson's disease is not solely psychological reaction to motor disability. It reflects circuit-level dopamine imbalance. Autonomic instability increases as degeneration spreads to brainstem nuclei. The dorsal motor nucleus of the vagus and other autonomic centers may be involved early, but clinical manifestations intensify as disease progresses. Orthostatic hypotension emerges when sympathetic regulation weakens. Patients may feel lightheaded upon standing due to impaired vasoconstrictive reflexes. Bladder dysfunction becomes common. Urgency and incomplete emptying reflect disrupted autonomic control. Gastrointestinal motility slows further. These changes are often underreported, but reflect progressive network collapse beyond voluntary motor systems. Sleep disturbances persist and may worsen. REM behavior disorder may evolve. Fragmented sleep, daytime somnolence, and circadian disruption reflect degeneration within sleep-regulating nuclei. Importantly, cognitive and autonomic symptoms often develop after motor threshold crossing, but may have roots in earlier phases. As nigral degeneration progresses, cortical dopamine tone declines further. Executive deficits become measurable. In some individuals, Parkinson's disease dementia develops when cortical and subcortical pathology becomes widespread. The progression beyond motor control underscores that dopamine network collapse is systemic within the brain. It is not a tremor disorder alone. It is a distributed degenerative process affecting multiple circuits sequentially. Motor symptoms may remain responsive to dopamine replacement therapy for years, but cognitive and autonomic symptoms often respond less robustly. This reflects structural degeneration beyond simple dopamine deficiency. When cortical neurons and additional neurotransmitter systems become involved, pharmacologic replacement has limited restorative capacity. The visible shaking hand is therefore only one component of a broader network failure. As degeneration spreads, the system regulating movement, cognition, mood, and autonomic function becomes increasingly unstable. Progression does not occur uniformly in all individuals. Genetic factors, environmental exposures, and baseline neural reserve influence rate and pattern. However, the underlying mechanism remains consistent: progressive loss of dopaminergic neurons. expansion of alpha-synuclein pathology, and destabilization of interconnected circuits. The early dopamine network collapse that began in the substantia nigra eventually reshapes frontal executive systems and autonomic regulation. The disease becomes less about tremor and more about global neural integration. Parkinson's disease is therefore not static after motor onset. It continues to advance structurally and functionally. The initial threshold crossing marks visibility. The subsequent spread marks systemic involvement. By the time cognitive slowing and autonomic instability are evident, the collapse has extended beyond motor loops. The dopamine network is no longer simply deficient. It is structurally fragmented across multiple brain systems. Irreversibility. Why dopamine replacement is not restoration. Levodopa improves movement in Parkinson's disease because it replenishes dopamine temporarily within the striatum. Tremor diminishes. Bradykinesia improves. Rigidity softens. The response can be dramatic, especially early in treatment. This improvement creates a powerful illusion that dopamine deficiency was the entire problem, and that restoring dopamine reverses the disease. It does not. Levodopa replaces neurotransmitter. It does not replace neurons. By the time motor symptoms appear, a substantial proportion of dopaminergic neurons in the substantia nigra are already lost. Substantial portion of their axonal projections into the striatum have degenerated. Synaptic terminals have disappeared. The structural network that once regulated motor circuits has thinned permanently. Levodopa enters surviving terminals and is converted to dopamine. It enhances signaling through remaining synapses. However, as degeneration progresses, the number of intact presynaptic terminals declines further. The capacity to buffer dopamine release becomes limited. Dopamine levels fluctuate more dramatically in response to medication dosing. This loss of buffering leads to motor fluctuations. Patients experience on periods of improved mobility, followed by off periods as medication effect wanes. The smooth modulation once provided by intact nigrostriatal projections is absent. Pharmacologic replacement cannot replicate physiological release patterns precisely. With time, dyskinesias may develop. These involuntary movements arise because striatal receptors have adapted to chronic intermittent dopamine exposure in the context of reduced neuroregulation. Postsynaptic supersensitivity combined with pulsatile stimulation produces abnormal motor output. Dyskinesias are not signs of excessive recovery. They are signs of network instability. Importantly, dopamine replacement does not halt alpha-synuclein aggregation or mitochondrial dysfunction within surviving neurons. The underlying degenerative process continues. Neuronal loss progresses gradually despite symptomatic improvement. Axonal degeneration is permanent. Long-distance dopaminergic projection fibers do not regenerate in a way that restores original network connectivity in the adult central nervous system. Gliatic changes within the substantia nigra and striatum stabilize damaged regions but prevent regeneration. As degeneration advances, non-dopaminergic systems become involved. Cholinergic, serotonergic, and noradrenergic pathways may also deteriorate. Dopamine replacement addresses only one component of a broader neurodegenerative process. Cognitive and autonomic symptoms often persist or worsen despite optimized dopaminergic therapy. Deep brain stimulation can modulate abnormal oscillatory activity within basal ganglia circuits. It can reduce tremor and rigidity significantly. However, it does not restore lost neurons. It adjusts network rhythm temporarily without reversing structural collapse. Irreversibility in Parkinson's disease is therefore anatomical. Neuronal death is permanent. Synaptic loss is permanent. Network thinning is permanent. Symptomatic treatments enhance remaining function, but do not reconstruct original architecture. The progression may be slow, but it is cumulative. Surviving dopaminergic neurons continue to experience oxidative stress. Alpha-Synuclein pathology may spread transsynaptically. Compensatory mechanisms weaken further as cell loss increases. The fear embedded in this stage lies in biological limits. Early dopamine network collapse began silently years before diagnosis. By the time tremor appeared, structural loss was already extensive. Treatment can restore chemical signaling temporarily, but cannot restore lost cellular infrastructure. Parkinson's disease is therefore not reversed by dopamine replacement. It is managed. The visible motor improvement masks ongoing degeneration beneath. The hand that steadies with medication does not mean the midbrain has healed. It means surviving neurons are being pharmacologically supported. The structural foundation continues to thin gradually. Irreversibility defines the disease. The collapse began before visibility. Treatment modifies output, not architecture. And once dopaminergic neurons are gone, they do not return. Stable aftermath. Parkinson's disease is commonly recognized by tremor. In reality, tremor is the late surface expression of a long silent collapse inside the dopamine network. The degeneration begins in vulnerable dopaminergic neurons of the substantia nigra. Mitochondria falter. Oxidative stress accumulates. Alpha-Synuclein misfolds. Synaptic terminals weaken. Compensation rises to masked decline. For years, the system adapts. By the time Bradykinesia appears, more than half of the nigral dopaminergic neurons are already lost. Striatal dopamine is critically depleted. Direct pathway facilitation has weakened. Indirect pathway inhibition dominates. Beta oscillations entrain motor circuits into rigid patterns. The visible tremor is not the origin of disease. It is the announcement that compensation has failed. Early dopamine network collapse is not a moment. It is a progression. First metabolic stress, then synaptic dysfunction, then increased compensatory firing, then structural neuronal death. Finally, network desynchronization and motor threshold crossing. Non-motor symptoms often precede motor collapse: loss of smell, constipation, REM sleep behavior disorder, mood changes. These are not incidental features. They are early signals that degeneration has begun in circuits beyond voluntary movement. Treatment restores dopamine temporarily. It improves motor output. It reduces tremor and rigidity. But it does not restore lost neurons. It does not reverse alpha-synuclein aggregation. It does not rebuild severed axons. Structural loss remains permanent. Irreversibility in Parkinson's disease is anatomical. Adult dopaminergic neurons are not regenerated in sufficient numbers to restore lost network architecture. Once axonal projections thin, they do not reconnect. Once network synchronization becomes pathologically entrained, flexibility diminishes. The fear in Parkinson's disease is grounded in timing. The visible symptom appears late. The degeneration started years earlier. The system was collapsing quietly, while movement appeared normal. By the time shaking begins, the structural foundation of motor modulation has already eroded. Parkinson's disease is not simply dopamine deficiency. It is dopamine network collapse. It begins silently in the midbrain. It spreads through basal ganglia loops. It destabilizes oscillatory timing. It extends into cognitive and autonomic circuits. It progresses beyond tremor. The hand trembles when structural thresholds are crossed. The slowness reflects inhibitory dominance. The rigidity reflects rhythmic entrapment. The non-motor symptoms reveal broader circuit involvement. This is pathology of fear, not because the disease is unpredictable, not because the mechanism is unknown, but because the brain can be losing the neurons that regulate movement long before the first tremor appears. And by the time the collapse becomes visible, much of the network has already been lost. And this fear is real.
Pathology of Fear is a medical documentary series presenting clinically documented neurologic and pathological mechanisms for educational analysis.